Thank you very much. So I named my speech. Can we start the presentation? Like I named my speech like this, but I'll be speaking about some other. This is just the modeling, a mathematical model of the DNA and deuterium influence on the open states occurrence. And at first I will speak about our biological experiments. So the aim of the work was to determine the biophysical process and mechanisms of deuterium-depleted water influence on the biological systems. So at first what we started, it was 2010, I think. We started from developing and constructing the plant to produce the deuterium-depleted water. And the next problem that is to check the concentration of deuterium in water and in biological fluids. So we didn't have isotopic mass spectrometer, but we used an NMR spectrometer. I will tell about this. And after that, we started to study the biological effects. And one of the last things we made, it was the mathematical model of DNA with explaining the mechanism how deuterium can incorporate in hydrogen bonds between nitrogen bases. Next. So this is the electrochemical plant with based on electrolysis with the metal electrodes, and here you see three of these plants that can produce in one section 50 ppm water. Okay, then the method to determine the concentration of deuterium in natural level and less. We used the shifting reagent, europium 3-fluoromethan sulfonate, and we use the ampoule with the water, and the other tube we put inside that consists with the europium. On the spectra we can see two separated signals, and with here we have we know the concentration here with the europium and this one, so we can count the concentration in the starting ampoule. So the same mechanism with O17, we can check the O17 concentration. This slide presents the deuterium decrease just in regular rats, V-star rats, with consuming water with deuterium content like this. I think many people did some such experiments, but here I want to show slides with deuterium content in kidney, liver, and on the in 5 generations. So this is the control concentration at the start of experiment. Then it is the first generation. We can see that in kidney and liver, in liver it's okay, but in kidney it didn't come to the level of the second, third, fourth, and fifth generations. And here, the heart, we can see the deuterium content here, and then the start of experiment. And the blood plasma. So in the control group, we have deuterium content. It is in delta promille, the deuterium content. So we can see that at first the DH gradient between blood plasma and tissues, the concentration of deuterium in blood plasma is much more than in tissues. But after consumption of deuterium-depleted water, the concentration of deuterium in blood plasma stood less than in tissues. And this can be one of the mechanisms of influence of deuterium-depleted water on the organism, because of the adaptation processes that can be during the changing the isotopic gradient from the at first the higher content of deuterium in blood plasma, less in organs, and then it's changed higher content of deuterium in organs and less in blood plasma. The next I wanted to show it's the experiment female that just born children. They drank water, deuterium-depleted water, and we checked the concentration of deuterium in blood plasma, in oral fluid, and in saliva, and in breast milk. And we can see that the deuterium content in breast milk is much less than in other fluids. And it's after the consumption, the breast milk still is less than the other fluids, the deuterium content. So the next one. The modeling of chronic antitoxicosis in rats. We have 5 groups, 6 groups. I will not stop on the explaining of every group. It's 42 days experiment, but the interesting things we could see, it's when rats started to drink deuterium-depleted water, and they got the model of endotoxicosis. This group was the worst in the experiment, and the best one group showed good results. Was the group that started to drink deuterium-depleted water 14 days before started to model endotoxicosis. And here the deuterium content is not too interesting in different groups, but the mass of the rats, a changing of the mass. So this one is the group that started to drink deuterium-depleted water and started the experimental to model the endotoxicosis. So we see the mass goes down much, and here this is just the control group. And this one is the group that got the deuterium-depleted water 14 days before modeling endotoxicosis. So we see it's near the control group that was just without any experimental change. It drank regular water. Okay. Another biological experiment, a rejuvenation effect that we saw on female rats, old female rats. You can see this is the control group, and the experimental group, the rats that that were drinking deuterium-depleted water. So we can see the replacement of yellow spinous hair with white, soft and shiny, as well as an also increase of motor activity of these rats. Yes, these were lazy and they didn't want to move and so on. But these were active, and after I think it was 28 days drinking the DV. And also the sexual cycle of these experimental rats stood near the young female rats. So we could see the partly changing the sexual cycle, and it was much better and near the young female rats.
This is a new Viagra.
Might be, but for females. Okay then. The mitochondrial experiment that Professor Lobushev said, that I think it was minus to me. We published the data and we wrote that the rats drank the deuterium-depleted water 28 days, but really it was a second generation of rats, and we took the liver and mitochondria from the second generation of rats in that drank DDW, and the control group was it's not DDW, this is just regular water. The control group was just the regular rats with the same age. So we saw the about 35% increasing of H2O2 production in by mitochondria from rats consuming the DV. The thing, some other experimental things that all the isolation of mitochondria from rats we made in the same water, in the same deuterium-depleted water, and mitochondria from regular rats in regular water. We had different combinations of like mitochondria from deuterium-depleted rat and the mitochondria, but isolation was in regular water and so on, different changing. But the most difference we saw here, just when the isolation was in deuterium-depleted water, and when the regular rats and isolation in regular water. Okay, then about open states in DNA molecule. So the sugar phosphate chains, and this like nitrogen bases. The open states we can see here, where where we can see the breaks between nitrogen bases. And the model, I will not stop on this, just Newton equations and different characteristics. So, but that show the different characteristics of the chain of sugar phosphate chains, like rotational dynamics, and this characteristics of nitrogen bases, like moving around the sugar phosphate chains, and so on. This is just the coefficients for each nitrogen bases, and experiments. Each line is one chain of the DNA. This is time, and this is the angle, and here is just 980 of nitrogen base pairs. This is just a smaller one than DNA. And this one without open states. And here we made the energy, disruption energy between nitrogen bases a bit less, and now we can see the open states, but it is better to see here as the same graph with open state, and this, so they can open, close, and then open again. And what for? To read the information, when the reading information from the DNA, there should be an open state, then and then starts the reading. Okay, like this. The energy that we took is you can see here newtons on meter. Next. Okay. The probability value of occurrence of open states. It looks like this. So this one is the one deuterium, and the probability of open states occurring. The concentration of deuterium in DNA we decided to take like the concentration of deuterium in the media. So the results I'll just show here. The probability of open states occurring grows when when deuterium content when we have just one deuterium atom on this 980 nitrogen bases, but this is not here. Just deuterium can be in any base pair, and this all counted, but we don't locate deuterium here. In other numerical experiments, we made the location of deuterium and showed that it is very important. And the deuterium location can influence on how many open states will be, name's bubble. The location of the one deuterium atom is very important, and we think that deuterium can get in the main place where it can play main role. So these are the bubbles where few open states here. So this is just nitrogen bases, and this is the time. Okay, then the conclusions. I think want to say that the breaking energy that we took and experimental data of Bockelman and others are the same, and the model that we used is very near the real, how it real works. So I think thank you, and I can answer any questions if there are some.